June 24, variant 1. As usual, we're going to read the question step by step. Cooking fruits and vegetables in water reduces their vitamin C content. The vitamin C is transferred from the fruits and the vegetables to the water by diffusion. DCPIP can be used to estimate the concentration of vitamin C. Let's draw it كده to understand. If you put مثلا an orange in water, okay, cook it in water, يعني hot water, the vitamin C in the orange will diffuse into the water. How can I test for vitamin C? If you want to test the water for vitamin C, how will you do it? Using the reagent, which is DCPIP. The blue DCPIP reacts with the vitamin C and becomes colorless. A student investigated the effect of. The word after effect of is usually the independent variable. I want to see the effect of cooking time. So I'm going to change the cooking time. This is the independent variable. I'm going to change the cooking time and check the vitamin C that diffused into the water. Okay, step number 1. A piece of foil was wrapped around the beaker. I got a beaker and I covered it with foil from the base and from the sides. I only left the top not covered. طب why do we put foil? الفويل على البيكر from the outside is always for insulation, so it doesn't lose the heat. A piece of citrus fruit was put into the beaker. The hot water was added. I added hot water to the beaker so that the piece of citrus fruit was covered. Piece of fruit was covered. And a stop clock was started. Okay. 1 cubic centimeter samples of the liquid. I took a sample from the liquid outside. Were taken every minute for 5 minutes. Every minute for 5 minutes. Each sample was put into a separate test tube. يبقى I take 1 drop and put it into 1 test tube. After another minute, I take another drop, I put it into the second test tube, third test tube, fourth and fifth. Every minute for 5 minutes. What did I do after? I got a syringe with DCPIP solution, 5 cubic centimeter of DCPIP. If there is any air bubble in the syringe, you have to remove it. You move the syringe so that the air bubble goes to the top and you remove the air bubble outside. Okay, 1 drop of blue DCPIP solution was added to the test tube containing the first sample. هنا. This is the first sample. I will add a drop of DCPIP. It is shaken for 5 seconds to mix the contents, and the color is observed. The student repeated step 7. I will continue to add drops until the volume of DCPIP, the color of DCPIP remains blue. دلوقتي this sample has vitamin C. So when you add a drop of DCPIP, it will turn colorless. طب I added another drop, colorless. Third drop, colorless. Fourth drop, it remained blue. يبقى I will record the volume of the DCPIP remaining in the syringe. Again, I will add the drops of DCPIP. I will add it drop by drop until it remains blue. This means that there is no more vitamin C in the sample. It's all reacted. So I would understand in إيه بقى? The more the volume of DCPIP used, the more the vitamin C in the sample. طيب, let's finish the steps. The syringe was refilled with 5 cubic centimeter of DCPIP And the steps were repeated with the rest of the samples, sample 2, sample 3, 4, 5, every minute. And these were the results. Every minute, I have a different sample. This was the volume remaining in the syringe. So can I calculate the volume added to the vitamin C? Yes. The original volume in the syringe was 5 centimeter. If the remaining is 3.6, يبقى how much was the volume added? 5 minus 3.6, it will be 1.4. 5 minus 2.8, 2.2. 5 minus 2.2, 2.8. Okay? Of course, using the equation, calculate this volume. He left this one empty. He wants you to calculate it. Originally, the syringe had 5 cubic centimeter of DCPIP. This is what is left. The sample that we took at 5 minutes, this is what is left in it, in the DCPIP. So how much was added? 5 minus 1.6, the volume added will be 3.4. 3.4. Prepare a table. Every minute, I had a sample. Okay? Record the results shown in figure 1.1 and your answer here. Every minute, I had a sample. يبقى on the left side, I will put the time. This is what you changed. Every minute, you took a sample. 1, 2, 3, 4, 5. The time of cooking. The volume remaining, I will put it here. And the volume added, I will put it here. Changed variable is always on the left. Results are always on the right. State a conclusion. Always remember that a conclusion is not a description. So you cannot just say as the time increases, the volume added increases. No, what does this mean? As the cooking time increases, more vitamin C diffuses out into the water. This is why it took more volume of DCPIP. يبقى you have to tell him a conclusion. What does this indicate? As the time increases, it's used a lot of DCPIP. طب what does this mean? It means that there is more water that there is more vitamin C that diffused in the water. This is the conclusion. Longer cooking time causes more vitamin C to diffuse out. What is the independent variable? Independent variable is the variable you changed. What did I change here? I changed the cooking time. Time of the fruit left in water. Cooking time, time of the fruit left in water. What is the purpose of the foil? We just said it. Why did we put foil around the beaker? To maintain the temperature, decrease heat loss, for insulation. Insulate the beaker. Explain why it was important to remove the air bubbles. As I told you, you have to shake the syringe so that the air bubbles goes out, because this air will take volume inside the syringe. And I want to put in the syringe 5 cubic centimeter of DCPIP. So if there is air inside, it will take part of this volume. To make sure that the volume of DCPIP is accurate. That was question 1. 8 marks for question 1. Let's see question 2. Plan an investigation to determine the effect of light intensity on photosynthesis in an aquatic plant. طيب. For this question, we have it in the plant nutrition chapter. This is an experiment that we have in our notes. I want to check the effect of light intensity. So I want to see how the light intensity changes the photosynthesis. يبقى this is my independent variable. On إيه بقى؟ Photosynthesis in an aquatic plant. So I have to measure the photosynthesis in some way. I will write my steps in order. Number 1, what are you going to get? What are you going to use in this experiment? I'm going to use an aquatic plant. One aquatic plant. What are you going to change? The light intensity. Light intensity. How? Using a lamp. You can put the lamp closer. It will be high light intensity. Then you can put it further away. It will be low light intensity. Number 3, what are you going to put the same? Anything else that affects photosynthesis. Temperature, the type of the water, the volume of water, the amount of concentration of carbon dioxide, whatever. Number 4, how are you going to measure your results? What are you going to measure and how? Photosynthesis of an aquatic plant. Plant underwater gives oxygen bubbles. So you can either count the number of bubbles or measure the volume of gas using a gas syringe. Number 5, 6, and 7. Improvement, safety precaution, repeat. For the improvement, you can maintain the temperature so that it does not change. Thermostatically controlled water bath. Safety precaution, you have a lamp, electricity, and you have water. So you can make sure that they do not touch each other. Then repeat at least 3 times and calculate an average. يبقى number 1, I will use one aquatic plant. What is the factor changed? Use a lamp, change its distance to change the light intensity. Same type of plant, same temperature, source of carbon dioxide. What are you going to measure? Volume of gas produced using a gas syringe. I can also say that I'll use a heat shield. What is the heat shield? It's something you put between the lamp and the plant to absorb the heat. Safety goggles, or keep the plant away, keep the water away from the electricity. Repeat at least 3 times and calculate average. Question 3, figure 3.1 is a photograph of a cross-section of a persimmon fruit. Oh. First question, he wants you to draw it. It's a very easy drawing. You just draw the outlines, but take care of any indentations or any projections. If there's a projection, I have to draw it. If there is a part that goes inside كده, indentation, I have to draw it. And then I will draw the seed holes, count them, draw them, and I will draw the seeds. 1, 2, 3, 4, 5, 6 seeds only. These have no seeds. Please do not color, do not shade anything, just the outlines. Outline the seed holes and the seeds. 1, 2, 3, 4, 5, 6. These two have no seeds. This is what I want from the drawing. Big drawing has to cover 50% of the space. Clear outlines and the details, the indentations and the seeds. After the drawing, there is the magnification question. Line PQ represents the diameter of the persimmon fruit. This is line PQ. You will measure it using your ruler. Your ruler gives a measurement in centimeter, but he wants it in millimeter. So the number that you got, you will multiply by 10. Multiply by 10. Calculate the actual diameter. Put your triangle. Magnification equals image over actual. I want the actual, so I divide the image by the magnification. Where is the magnification? Under the picture. 104 over 2.2. It will give me a lot of decimal places. He wants three significant figures. So 47.3. 47.3. Okay. The plants can make fats, starch, and proteins. State the names of the reagents that can be used to test the samples of the plant tissue for starch and protein. Food tests are very important. Uh, what is the reagent for starch? Iodine solution. What is the reagent for protein? Protein biuret solution. Starch is iodine, protein is the biuret solution. How can I test for fat? The fat is the emulsion test. You get the fat sample and you put it in alcohol, ethanol. You add water and you shake. Shake is important. It gives you the second mark. You have to shake. If it turns cloudy, there is fat. If it's not cloudy, then there is no fat. Okay. Last question. Another experiment. Pectinase is used to produce apple juice. It breaks down the cell walls, so it increases the volume of juice that can be extracted. If you use pectinase, it will give you more juice. The student investigated the effect of a concentration of pectinase on the volume of juice extracted. So what am I going to change? Anna, I want to see how the concentration of pectinase affects the juice. يبقى this is my independent. I will change the concentration and measure the volume of juice that I got. And this is my dependent variable. The masses of 7 samples of chopped apple were measured. They were put into different beakers. 50 cubic centimeter of pectinase was added to the 6 beakers. يبقى same volume of pectinase. But the concentration was different. This is what you changed. They were kept in a thermostatically controlled water bath, 37 degrees Celsius. The contents of the beaker for 30 minutes, all of them for 30 minutes, and the same temperature for all. Contents of the beaker were filtered and the liquid was collected. Volume of liquid was recorded. What is the dependent variable? What were the results you were measuring? I was measuring the volume of liquid at the end, اللي هو الجوس. Volume of juice at the end. This is my dependent variable. طب what were kept constant? I have a lot of variables kept constant. The temperature, the time in the water bath, the volume of pectinase, anything. Identify a hazard. What is the hazard? Something dangerous. He chopped the apple. Chopped them, yani he cut the apple using a knife. So this is the hazard, cutting the apple. How can you protect yourself during cutting? You have to cut on a hard surface or on a cutting board, not in the air. The student did not repeat. Amala, he only did the experiment one time. Why is it better to collect several sets? It is better to collect several sets. It is better to repeat. Why? Every time he asks you about the repeat, we say two things: increase reliability, identify anomalous results. Why do we repeat? To identify anomalous results and increase reliability. The seventh beaker of chopped apple did not contain the pectinase solution. What should have been added to make it a control experiment? I removed the pectinase. What should I add instead for a control experiment? I can add water. I can add the same volume of boiled pectinase or the same volume of water. The results of the investigation is shown. This is the concentration of pectinase, and this is the volume of liquid collected. Gram, this is the volume of liquid collected, cubic centimeter per gram. Each gram of apple gives this volume. Each gram gives this volume. Okay. What is the question? A sample of 150 grams of apple was placed in a beaker of 0.8 pectinase. Use the table to calculate the volume of liquid collected. طيب. In the table, he told you that at this concentration, 1 gram of apple gives 0.92 juice. At this concentration, 1 gram of apple gives 0.92 juice. So if you want to calculate 150 grams of apple, cross multiply 150 times 0.92 divided 1. It will give you the volume of juice, 138 cubic centimeter. Plot a line graph on the grid. When he asks you for a line graph, you plot your points and then you join point to point using a ruler. Okay, what are my rules? Left side of the table is on the x-axis. This is the x, this is the y. The results you were measuring is on the y-axis. First step, put your labels with the units. Put the full labels with the units, here and here. And then I'm going to put my scale, 0.24681. I have from 0.2 till 1. 0.24681. On the y-axis, I have from 0.69 till 0.92. يعني I can put a scale from 0.65 to 0.95, for example. I don't have to start before this, and this is what I did. 0.60 or 0.65, 60, 65, 70, 75, 80, 85, 90. Yes, 90, 92 will be here. Or I can start from 0.65 also. It would be fine. Now let's put the points. 0.2, it was 0.69. Type as the 0.2, this is 65. Each two boxes is 0.01. يعني this is 0.65, 66, 68, 66, 67, 68, 69. Oh, and then 70. Each two boxes is 0.01. 0.4, it was 0.77. 76, 77. And then 81, 81. 85 92 92. لا 1 second احنا عندنا 0.2 0.4 0.5. 0.5 is the 81, not the 0.6 here. Okay. 0.6 85, then 92 92. 0.6 85, 92 92. This is the 90 91 92. There you go. What will I do after this? Join point to point using your ruler. Point to point. Point to point. Okay? To make sure you don't get it wrong. This would be my graph. The last question, describe the relationship. This is a description, not a conclusion. As the percentage of pectinase increases, the volume of liquid increases, and then it levels off. As the concentration of pectinase increase, the volume of juice you get will increase, and then it remains constant or levels off.